We have Professor Alex Lipka, who's going to give us a talk today. He's a professor of biometry in the Department of Crop Sciences at the University of Illinois at Urbana-Champaign in Chicago, and is the PI of the Lipka lab. They apply cutting-edge statistical approaches to quantitative genetics analysis for crops. So I'm just seeing other people in. And this research results in more accurate quantification of genomic signals, underlying phenotypic variation, and prediction of breeding values for agronomically important traits. Professor Lipka's main research interests involve incorporating functional biology, breeding, and molecular mechanistic knowledge into everyday quantitative genetics and statistical models. So for this talk, we'll focus on quantitative genetics, which is critical for understanding how and to what extent genomic variability contributes to observed agronomic trait values. So Professor Lipka will be providing two lectures, one today and one tomorrow. And just for some house rules, the webinar is being recorded. Please keep your microphones on mute, and you can type questions in the chat or you can raise your hands and ask yourself questions. So thank you so much, Professor Lipka. Over to you.
Okay. Well, thank you for the introduction. And I just want to say it's going to be an honor to be able to present these slides for today and tomorrow to you all. And I view this as more of a conversation than as a lecture. So please, um, ask questions. You can ask questions during the presentation. You can type questions into the chat. And I asked Sumir to help me out. If anybody's asking a question and I'm just not realizing it, I asked Sumir will help me out making sure that your questions are asked, okay? So let me just minimize my workspace over here. Okay, and okay. So I have about 30-ish slides, maybe slightly less. So I view that this will be sufficient to take up the hour. And what we're specifically going to talk about today is obtaining best linear and biased estimators and best linear and biased predictions from multi-location experiments. So if you're growing, crops in multiple locations, multiple years, blues and blups are a way to get one summary phenotypic measurement for your genotypes that you can bring forth for genome-wide association studies and genomic selection. Today we're going to cover an introduction to the genome-wide association study. And then we're going to do a basic example of a GWAS in maize. All right. Um, so let's say we have a multi-location experiment. So for example, let's say we have seeds obtained from a germplasm bank. Okay. So you got these seeds that are being planted, and then you come up with a panel, breeding material or diversity panel, right? And they're plotted and they're planted in multiple replications in multiple environments. So then the question is, how can we summarize this trait information across multiple reps and locations? All right. So what we're going to do is we're going to fit a statistical model that will account for these multiple sources of trait variation. You know, your genetic effects, your environmental effects, your G by E. And then the output from this model are going to be either BLUPs or blues of trait values for each of your genotypes or your taxa, your accessions, whatever you want to call them. So are there any questions so far? Okay. Okay. I don't see any, so I'll keep on going. Um, so a quick review of fixed effects versus random effects. So let's say we have an experiment. We have a factorial design. Let's say factor A is density, like how closely are the plants planted together in a field. And let's say we have three different levels of density, okay? And then factor B is fertilizer. How much fertilizer are you going to apply? Okay, so let's say that there are three different levels of fertilizer. So there's going to result in 3 times 3, 9 possible combinations of factors A and B. And so then let's say we randomly assign 27 plants to each combination of A and B. So therefore, we have this 3 by 3 factorial design with 3 replicates per each combination of factors for each treatment level. And let's say we measure a response variable. Let's say we measure yield on all 27 plants. Okay. So in our ensuing statistical models, we will have um, a main effect for factor A. So this is looking at how does the mean yield differ across each of the 3 density levels. And then we quantify our main effect. And then we have factor B, how does the mean yield change across our 3 different levels of fertilizer? And we quantify the main effect of factor B. And then we look at our interaction effect of factor A and factor B. So knowing something about the level of factor A is going to tell us about the yield of a particular level of factor B. So we then quantify this two-way interaction effect. And the way we quantify these main effects and interaction effects is through this statistical model, okay? And where we're going with this is we're going to get more and more and more specific until we end up with these blues and blubs that I was talking about. So why IJK? That's the yield of our kth replicate receiving the jth level of factor B fertilizer and the ith level of factor A. Then we have our grand mean. Then we have our fixed main effect to the ith level of factor A, our fixed main effect to the jth level of factor B, and then our two-way interaction effect between the jth level of factor B and ith level of factor A. And then we have our random error term, which is assumed to be normally independently distributed, population mean 0, population variance sigma square E. So here we're treating all of these as fixed effects, right? So each of these effects, these main effects and two-way interaction effects, these are parameters that get estimated through your data. And the take-home message of this fixed effect is that the inferences you make in such a model, they technically only apply to the factor levels in your experiment, right? So our inferences will apply only to our 3 particular plant densities and our 3 particular fertilizer levels. All right? Now let's contrast this with the same model, but we're going to make our main effects and interaction effects random, right? So the y variable does not change from the last slide. Grand mean does not change from the last slide. But uh-oh, look at this. So our random main effect, we're no longer assuming this is a parameter that gets estimated, but instead we say that this is a random variable that's normally distributed, population mean 0, population variance sigma square A. Similarly, if we assume that the main effect of factor B is random, right, then we're going to assume this is a normally distributed random variable. And same deal for this two-way interaction effect. Okay? And then the inferences we make from this random effects, since we're assuming that the main effects and the interaction effects are random, we then say that the inferences from this model apply to an entire population of factor levels. So, not only does this apply to our 3 particular plant densities, but it applies to all plant densities, right? And then for factor B, we can make a similar argument that our inferences will apply to all fertilizer levels. So are there any questions? Okay. And then a mixed model has both fixed effects and random effects, right? So let's just run through this model and see what changes, what does not change. Okay. So our y variable does not change. Our grand mean does not change. And let's say we want to keep, me as the experimenter, I want to say that I care about these 3 particular plant densities, so I'm going to have it as a fixed effect. All right. And then I want to say, I don't care about these 3 particular fertilizer levels. I care about all possible fertilizers. Then I might want to make the jth level of factor be a random effect. Okay. And then, if you have one main effect that is fixed and one main effect that is random, what happens to the interaction term? Well, remember the saying, okay? Random wins. So if you have one fixed main effect, one random effect, the corresponding two-way interaction effect is going to be random. All right. And then we have our error term. And mixed models, I really like them. They are extremely flexible and they can be adapted to many different quantitative genetics analysis. Okay. So not only are they going to apply to what we're going to see in the immediate future, um, to calculating blues and blups across multiple locations, but they also form the basis of statistical models for GWAS, and I would argue through like the GBLUP model, which we'll learn about later on. They also apply to genomic selection. So mixed models are really useful in quantitative genetics, and we use them all the time. So let's now put this basic theory of a mixed model into the storyline of we want to obtain blues or blups of our genotype effects across multiple locations. Okay, so we'll then say this is the phenotype of the kth replicate in the jth environment of the ith genotype. We then have, if we want to obtain blups of our genotype, we will have our genotype, our main effect of our genotype as a random effect. All right. And then, you know, again, our objective here is to obtain blups of our genotype. So I would argue it does not matter as much if you want to have your if you want to have your environment as a random effect or a fixed effect. In my experience with running these models, more often than not, I see the environment effect being included as a random effect. Our G by E effect is random, and then we have our grand mean and our random error term. So what we get from this is our blups of our genotype effect, and those blups can be carried forward to do GWAS or genomic selection or any downstream quantitative genetics analyses. Another thing that we get that's really important is um, variance component estimates for our genotype effect, our environment effect, our G by E effect, our error term. And those can be used to calculate heritabilities for us. All right. Um, so any questions here? Okay. All right. So now let's talk about obtaining blues. So we have pretty much the same model, right? The only difference is that your genotype is a fixed effect, okay? And then we keep our environments random, we keep our G by E term random, and our grand mean and random error term does not change. So what we get out here is our blues of our genotype effects. All right? And I'm getting a lot of emails. That's why you hear those noises. So, what should you use, blues or blups? Well, let's go over the advantages and disadvantages of them. Okay. So, blups, I would argue that blups make sense from a biological perspective, right? So, I would argue that if you have a data set of accessions or taxa or lines, I would guess that one would want to make inferences not to only those particular lines, but to an entire population of lines. Like, I don't know, all the relevant germplasm in the local area that you're growing, right? So I think BLUPs make the most sense from a biological perspective. However, a disadvantage is that BLUPs shrink values towards the mean. So what do I mean by this? Well, as a random effect for your genotype, okay, um, you're always going to systematically underestimate the value of your genotype. You're going to systematically underestimate the phenotypic value of that genotype, okay? So, such underestimation, you can think of it as shrinking the values towards the mean. So that's not advantageous, right? And then also another disadvantage is that fitting random effects is more computationally intensive than fitting fixed effects. So if you fit a fixed effect in a model, you have kind of most of the time like a closed form solution, like least squares estimates for obtaining your parameter estimates, right? For random effects, you have to do usually more iterative algorithms that converge to a solution. So that's more computationally intensive. Um, Blues. A main advantage of blues is that since they correspond to fixed effects, they do not shrink values towards the mean. So what you see is what you get. Another advantage is that they're less computationally intensive, right? So iterative algorithms is not required to estimate the parameters. Um, a disadvantage is that technically, if you're including your genotypes as a fixed effect, then the inferences you make only apply to those particular genotype levels. So I would say blues do make less sense from a biological perspective. All right. Are there any questions? Okay.
Yes, please.
Oh, yes.
Okay, good day. I just wanted to ask again, please, could you re-explain what you mean by biological, like from a biological perspective? I'm not very clear on what that means.
Okay, so the question is, what is meant from a biological perspective? Yeah, so this is what I meant by that statement. So let's imagine that you have your germplasm that you're growing out in the field, right? Um, so so I don't know, you might have, I don't know, say like 100 accessions being grown in the field, right? Um, I would argue. I mean, please correct me if I'm wrong, but it would make sense to say that what I want to estimate in my quantitative genetics analysis does not apply to just these 100 accessions in the field, but it would apply to all possible accessions that I would want to plant, even the ones that are not in my experiment. Um, so to me, from a biological perspective, since I would want to make inferences to a larger group of accessions than just the ones in my data set, then um, Lux would make more sense. Does this answer make sense?
Honestly, I think this is like, I'm not very experienced in this field at all. Like,
Mhm.
honestly, I'm just here to learn. So, I mean, I understand it better than I did before, but genuinely, I think I just have a lot of personal research to do. But thank you for taking out the time.
Oh, of course. And please continue to ask questions, okay? Everybody, I love answering questions, especially the ones that I don't know the answer to. So yeah, so
Question?
yes.
Yes. I my question is, assuming, for example, you have an experiment
Hmm.
and you have some missing values,
Hmm.
and then maybe when you have those missing values, you want to use these blues or blabs, but
Hmm.
then the experiment also maybe may have experienced some, let's say, stress.
Hmm.
Um, Which one would you advise to use? Because is it the blues or the blacks?
Yeah. Okay, so let me describe this situation. So let's say we have an experiment and
Yes,
some of your accessions or taxa die because of stress. And so you cannot obtain a phenotypic value, right?
yes,
And so you're asking, should you use blues or should you use blups? Is
yes,
Yeah, so I think so. If you asked me this question 10 years ago, I would say it doesn't matter. Use the one that you're more comfortable with. Um, Now I am a firm believer that blues should be used. Um, and the reason why is this shrinkage thing, okay? When you're doing GWAS and you're doing genomic selection, um, you have to include your genotype effect as a random effect. Okay, so you are, like it or not, in the GWAS or genomic selection phase, going to have to shrink your estimates, your predictions. So you are going to have to underestimate the importance of each genotype. So I would argue that in this phase of the analysis, absolutely keep it as a fixed effect. Therefore, use blues. So you will not, you know, have double shrinkage. Does that make sense?
Yes, thank you very much. Okay.
Yeah. What an excellent question. Um, and actually, I'm about to point you to a paper that just came out. Well, I think it came out December 2024, so it's not very recent. But about this very subject. But are there any more questions? Okay. Well, I want to make it clear.
Sorry, sir.
Oh,
Sorry.
yeah.
Thank you, Alex. Did you say since they were missing values, we should go ahead with blues? I didn't get that part. But you was.
Yeah. So go go ahead with blues. Okay. Um, so you will not shrink your genotype value. And And I think I just used too much jargon in that answer. So I'll take a step back, right? So we want to make sure that we do not unfairly underestimate the true effect of a particular genotype or a particular taxa, right? Um, and so the way to make sure we do not do that is we use blues instead of blups.
Okay,
Does
thank
that
you.
answer? Okay. You're welcome. And see, I feel so strongly about preferring blues over blups that I literally made a slide about this. So I now prefer blues over blups. Okay. So, kind of answering this really important questions, or these important set of questions. Blups shrink your values towards the mean, right? So this means that genetic effects are estimated or predicted to be closer to the mean than they actually are. So this means that we're underestimating, or technically for random effect, under predicting the genetic values, right? So then the second problem is for GWAS and genomic selection, we need to include the genetic effects as random. Okay? Um, so we have to use blups in the GWAS and genomic selection phase, essentially, just to slightly oversimplify. And so the solution is the title of this paper from Hans-Peter Pifo and Jim Holland. It's called Don't Blup Twice. So take blues of your genotypes in this previous phase I was telling you all about, okay? And then you're only going to have to deal with shrinkage, that is underestimating your values of your genotype values only in the second phase of the analysis. Um, and let's see, so this is being recorded, so you can easily see this DOI over here. I strongly suggest reading this paper. It's a shorter paper, so it's not that long of a read, but it really goes through why you should get blues in the first phase where you're summarizing phenotypic values across locations and environments. So any questions? Okay. Well, I'm really appreciating the questions and the conversation we're having so far. Mm, okay, some really quick technical notes. In plant breeding, you add the estimate of the grand mean to your blues and your blips. Okay, and we still call them the blues and the blips, which is one thing that confused me a little bit when I jumped from applied statistics to quantitative genetics. And the reason why we add the mean back is that the blues and the blues will be in the same units of measurement as the raw trait data. Um, I would still call them blues and blobs. Um, Also, another thing is that your statistical models follow assumptions, right? So always consider transforming your Y variable before fitting statistical models. And this will help out with, the assumptions of normally distributed error term and constant variance. Um, so I gotta make a confession. I now run a lab, so I'm usually not the person fitting these models anymore, but I used to be. So my lab uses ASRemal and R, and for historical purposes, I'm going to include SAS, but I haven't renewed my SAS license since like a decade. Um, an advantage of ASRemal is that it can fit very complicated models with a lot of random effects very quickly. A disadvantage is I would argue that there is a learning curve to it. It's not simple to use. And then the second disadvantage is that there's an expensive annual license fee. Like I believe that you still purchase it out of the UK and it's like £500 per year or something like that for researchers. Um, an advantage of R is that it's free. The disadvantage is that it's not as extensively tested as SAS or ASRemal. So there might be errors in the code. So definitely, you know, do a thorough inspection of the results and like pressure test what you get. SAS, um, it's relatively simple to use. A disadvantage is that there's an annual license fee and it's slow. Like if you have a lot of random effects, SAS will take forever. And I really don't use SAS that much anymore. Are there any questions here?
Uh, Hi, Alex. I'm enjoying the lecture. Just a quick comment.
Mhm.
So for those, like I had someone asking if you have missing data points,
Mhm.
or if your experiment is starting from incomplete block designs, for instance, or yeah. So assuming you're starting with blobs already, because
Mhm.
some people advise if you have so many missing data points, it's better to kind of estimate blobs.
Mhm.
So if we already have blobs already, so what I do is I deregress the blobs to I deregress the blobs to kind of use in genomic prediction so that I don't have the double shrinkage thing you're talking about. What's your comment on that?
Yeah, deregressing. Yeah, so I will be honest. I have never really de-regressed things myself. So let me just take a step back and talk about what that is. So de-regressing, that is where you have like a blip and then you kind of turn it back to its original units of measurement in separate environments. Is that correct?
Yes.
Okay. Yeah, I think that that is a very useful strategy to deregress. And again, I have not personally done that before, but I think that that's a good way to, you know, have your cake and eat it too, right? So you can um, bring it back to your original units of measurement. So you have the original measurements in separate environments, and then you can calculate blues, and then you won't have double shrinkage. Yeah. Yeah, thank you, Nelson. I see Cool Deep has their hand raised.
Yeah, thank you, Alexander. So continuation to this, I have just like couple of doubts. First is
Mhm.
like when you are working in a hybrid crop, the line and tester, basically male and female effect also need to be quantified in your genomic selection. And
Mhm.
suppose we have almost 5 to 6 years of our regular trialing data, like stage one trial we used to have, which where we usually have a tester which represent the opposite pool and then we cross the parents. So I'm just giving you the context. So my question, there is always a double shrinkage and there is always a discussion about whether to go for two-stage blues and then use that two-stage blues to model your genomic selection. So what's your thought when we have almost 5 to 6 years of data, multi-year, multi-season data,
Mhm.
representing our maybe 3 to 4 zones, and we just want to pull it. So in that situation, going for two-stage blues is fine, or then just having blobs and then going for using those blobs to have male and female interaction effect, and then you predict for your hybrid.
Hmm. Yeah. Okay. So
I
so
think
this
which
is a
could be the
very,
best strategy where I could able to get.
yeah, yeah.
Please, go ahead.
So okay, so let me make sure I'm on the same page. So here we're talking about hybrids, right? So you have, general combining ability of both of your parents and then the specific combining ability, right? And then on top of that you have um multiple field seasons, like you said, 5 or 6 field seasons. And so the question is, should you still calculate blues instead of blups? Is that the question?
Yeah, which could be the better option to shall we go for blobs or blues? Because we have maybe almost 5 6 years of data. Like you can consider as a 5 to 6 seasons across almost 7 locations, almost 50 plus trial, and it's a huge
Yeah,
metal representing our two genetic pools. So there is always confusion. Shall we go for blobs or blues? Because sometimes you have missing data also. You can't able to get the all hybrid data. So then it's better to go for blobs. But when you go for blobs, it will be a shrinkage, specifically for trait like
yeah,
maybe flowering and all this. For we have very less variance effect. So
yeah,
that's how like how to deal with such kind of situation. So that's my
yeah.
question.
Yeah, yeah, yes. So that is definitely a complicated situation where you have multiple years with potential missing data, and plus, you're dealing with hybrids. Um, Now, I have not seen your specific experiment, right? So, don't spend the next year doing what I'm about to suggest, but my gut instinct is to calculate blues. Like you do, like, yeah, so maybe I would fit a model that has like a genotype effect in it, um, so that I can get blues or a single blue of your hybrid genotype across all environments. And essentially that would factor out the environmental effects, right? And then with your blues, which have no shrinkage to them, that's the thing where I would, in the next phase of my analysis, look into like the general combining ability of the parents and like the specific combining ability. Um, that's my gut instinct.
Yep. Thank you.
Mhm.
Thank you. Thank you.
Okay, great. So let me just peek ahead. Okay, now we're going to start talking about GWAS. So are there any final questions on the subject of blues versus blups? Okay, so on to our main event, the genome-wide association study. Okay, so the purpose of a GWAS is to identify genomic regions associated with the genotype. Okay, so at each SNP across the genome, we're going to fit a statistical model, and then we're going to use the fitted models to test the null hypothesis of no association between the SNP and the phenotype. Um, so a genome-wide association study finds regions with strong statistical association with your traits. Okay, so here is a study I did that was published way back in 2013. Our trait of interest was vitamin E levels in maize grain, and here on the x-axis we have genomic position of chromosome 5 in maize. And then the y-axis is our negative log base 10 p-value for this hypothesis test conducted at each SNP. So the taller the negative log base 10 p-values are, the stronger the evidence there is against this null hypothesis. So in this particular analysis, which I'm going to show in detail a little later, oopsie daisy, we have a peak association of a SNP within a gene that makes biological sense. Um, So this is an example of a success story with GWAS. So then markers exhibiting peak associations are potential targets for marker-assisted selection. So what can a GWAS do and what can a GWAS not do? Um, a GWAS cannot biologically validate or confirm the presence of a gene having an effect on your trait. Okay. What we're doing here is a statistical analysis. Okay. Peak associated markers are not, slam dunk evidence that you identified a causal relationship of your DNA to your RNA to your protein to your metabolite to your trait, right? It's just a statistical association. What a GWAS can do is guide future research, okay? For example, maybe it can help pinpoint targets that somebody would want to try CRISPR out on, or, QTLs for breeding. Um, and also maybe it can help, identify a target for which you want to characterize the surrounding linkage disequilibrium. And then also it can be a starting point for performing additional statistical analyses to refine the degree of statistical association with your trait. Any questions here? Okay. So a lot of the times when we do a GWAS, we use an association panel or a diversity panel. And what this is, it's a collection of a diverse set of germplasm from many different subpopulations of a species. Okay. Um, so here's an example from the so-called Ames panel, in the collected in by USDA researchers in Ames, Iowa, United States. So here we have these 2,815 maize lines, okay? And then our x-axis is principal coordinate one. So a principal coordinate analysis is very similar in spirit to a principal component analysis. And then our y-axis is principal component two. And so what we're seeing is that various subpopulations based off of, oh, I forgot to mention, this is calculated, this principal coordinate analysis is calculated based off of genome-wide markers. So what we're seeing is that the markers are essentially clumping all subpopulations together, okay? So if we do not account for these false positives, we may not be able to find true, signals that are near genes actually controlling the trait. So there are two sources for false positives, population structure, which we're seeing over here. Then another source is familial relatedness, right? So, the best explanation of familial relatedness I've heard was, let's say that you sequence my DNA, okay? And then I have a missing genotype at a SNP. And then you sequence my sister, okay? And she has that marker not missing. And then you sequence a random person walking down the street, okay? And they have their marker genotypes measured as well. Well, familial relatedness says that, using my sister's DNA is a better guess of what's in my DNA than a random person walking around the street. So that's familial relatedness. So we use a mixed model to reduce false positives in GWAS. And I think that this model, this mixed model, is the most statistically rigorous way to conduct a GWAS. So we have our phenotype of our ith individual. We have, for a particular SNP, we'll include it as an explanatory variable, where we have an allelic effect substitution, alpha over here. And so if you're doing a GWAS of, say, 1 million SNPs throughout the genome, you're going to fit 1 million of these models, one for each SNP. And then to account for fixed effects, to account for a population structure, my bad, you're going to, include these fixed effects, for example, principal components, all right? And then to account for familial relatedness, you're going to include line as a random effect, okay? And then you're going to have, as part of the variance covariance between your lines, you're going to have this kinship matrix to measure the relatedness between individuals. And then, of course, you have your grand mean and your random error term. So this is the best model you can use for a GWAS, in my opinion. So estimating your variance components are computationally intensive. So there are a lot of computational algorithms out there to make things more computationally tractable. I'm going to go over 3 of them. So the first approach we can do is a compressed mixed linear model. So what you do is you perform hierarchical clustering on your taxa, your lines, your sessions, okay? And then you use that hierarchical clustering to toss individuals into groups, okay? So then you replace your line random effect with a group random effect. And this speeds up computational time because you're working with a smaller kinship matrix and all of the underlying linear algebra operations are quicker. Another approach is population parameters previously determined, P3D, which is the same thing as MX, maybe if you've heard of that. So what you do is prior to running the GWAS, okay, you're going to fit a model that does not include this alpha xi term, and you're going to estimate the variance component for your groups or your lines, and you're going to estimate the variance component for your error term, and then you run your GWAS with these estimates, okay? So what that means is instead of running computationally intensive operations, 2 million times for each SNP, you're only doing it once. And that's going to substantially reduce computational time. All right. So, however, a thing about this is that this is an approximation. So we want to have statistically optimal estimates of these variance components, right? So with P3D, you get something really close to the statistically optimal estimate, but you don't get the exact optimal one. So approach 3 gives us the statistically optimal estimates. And what it does, behind the scenes, it replaces the computationally intensive calculations with the less computationally intensive calculations. So it calculates stuff only once instead of once for every step. And then we get statistically optimal variance components obtained at each marker. So you had the same substantial reduction in computational time as the approach I previously showed, but it's exact. It enables statistically optimal estimation of marker trait associations. Now keep in mind, I told you how all of this works. With software, this does this for us automatically. So when you're running a GWAS, you don't have to know the ins and outs of this. You just need, just be aware of it. So what are we talking about next? Yeah, okay. So any questions before I talk about the software that my lab uses for GWAS? Okay. So my lab uses 2 different softwares to run GWAS. One is Gapit, and an advantage of Gapit is that it automatically produces tables and figures summarizing results. A disadvantage is that I think that Gemma, which I showed in the previous slide, is the optimal GWAS approach. It does not use Gemma. Um, so the other software my lab uses for GWAS is Gemma. And an advantage of this is that it uses Gemma, right? I'm getting so many updates. And then a disadvantage is that it does not automatically produce tables and figures summarizing results. And the second disadvantage is that it requires more of a learning curve. All right. So any questions here? Okay. And oh, sorry, Nelson, yes.
No, I was just reading someone was asking, is it Akani?
Is it, sorry, I didn't catch that word.
There's someone who's asking a question in the chat.
Mhm.
What is the number of genotypes required to conduct a GWAS study?
What is the number of genotypes required to conduct a genome-wide association study? Um, it depends, okay? It depends on the genetic architecture of the trait. Um, I would say off the top of my head, minimum of 500 for a complex trait. However, I have run GWAS for simpler genetic architecture traits with sample sizes as small as 200, and I think even less than 200. Um, so that was my long-winded answer. Short answer off the top of my head to give a ballpark estimate, I would say minimum 500. Okay. So how to choose the best model for GWAS? Okay. So there are a lot of GWAS models out there. Short answer is use the unified mixed linear model. But here's kind of an um, underlying rationale. Okay. So it is critical to account for population structure and familial relatedness in a typical GWAS. All right. Um, Hands down, unified mixed linear model is the best option. Okay. Um, Now, there have been developments in the unified mixed linear model that can make it handle more situations. So, for example, there's a multi-trait unified mixed linear model. So your Y variable is a vector of traits instead of just one trait. There's a multi-locus mix model that can handle or that can do stepwise model selection for selecting multiple particular loci associated with the trait. And then there are other models that can account for non-additive effects and can account for unequal variances across genotypes at a particular SNP. So um, and then multi-locus, multi-trait models that include multiple traits in one model and multiple loci. So here's my suggested strategy, okay? I think you should always try out multiple statistical models in any statistical analysis. So as a first pass, use the unified mixed linear model, and then if and when you find something interesting, you know, try running more tailor-fitted statistical models um, to refine, and dig deeper into the associations that you're quantifying. All right? Okay. All right. So I do not have a clock in front of me, so I don't know how much. So I think we have maybe about 13 minutes left or so. So I'm going to go through this example, okay? I'm confident I'll finish it in enough time for there to be to answer questions. But please do stop me if there are questions, all right? Yes.
I just wanted to draw your attention to the chat where there's a couple questions and comments.
Oh, thank you for that. Okay, let's go to the chat. Okay. Okay, thank you. Okay. So I already answered what is the minimum number of genotypes required. What about the minimum number of markers? I think it's going to depend on your species. Um, and I would characterize the linkage disequilibrium in your panel, and I would study this on a case-by-case basis to determine the minimum number of markers. And then in general, okay, if you had a choice between increasing number of individuals or increasing number of markers, you want to always increase your number of individuals, okay? So you can have as many recombination events observed in your panel as possible. So if there's a question of more individuals versus more markers, always choose more individuals. Okay, so then there's a question when to use GAPIT or GEMMA. The both methods use the same requirement. I would say GAPIT or GEMMA are both acceptable because they're both using the unified mixed linear model. Um, I would say GAPIT has less of a learning curve. And then Gemma has a little more of a learning curve, but it's a slightly more statistically optimal estimate. So both are acceptable and both, if used properly on properly quality controlled data, should do well. Okay, Professor could explain a bit the linkage disequilibrium principle. Yeah, so linkage disequilibrium, right? So we can think of linkage disequilibrium as like, you know, two markers that are close to each other, right? So they are more likely to appear together in a haplotype block than markers far apart. And that was a really oversimplified definition of linkage disequilibrium. So um, markers that are closer to each other are more likely to be in the same haplotype block, right? So linkage disequilibrium is how we can measure um, genetic distance from each other. So yeah, I can post a DOI, the blood versus blue paper. So basically, you want to make sure that you have linkage disequilibrium present between your SNP and your genotype panel, okay, and nearby genes that may not be captured in your marker set. Okay, so it is hoped that your SNP that you're measuring in your GWAS has sufficient linkage disequilibrium with your neighboring genes, okay? Because usually your SNPs are not causative, right? So you want to make sure that you're tagging gene effects. All right. So that in a nutshell is a linkage disequilibrium principle. Could the DOI of the blood versus blue paper be put in the chat here, please? Yes. What if, how about I go through the rest of these slides and then before I end, um, someone remind me if I forget, I will post the DOI to that blood versus blue paper. Does that sound good? And then could the slides from all lectures be shared, please? Yes, I'll be happy to share them. And please feel free to follow up over email. And I can absolutely figure out a way to get these to you. All right? Okay. Um, okay. So let's go over this example. So we have a 281 member maize diversity panel. They were grown at two locations and quantified for these vitamin E compounds called tocochromonols in the seeds. Um, and so then our colleague at Michigan State, Dean Della Penna, went ahead and measured tocopherol levels in maize grain. And then and then we fitted a mixed model to account for field season effects, fitted each phenotype. And then this was before I saw the light about using blues. So we calculated blubs. And then we had 20 tocochromonol compound sums, ratios, proportions that are analyzed in GAPIT. And then we used about 294,000 SNPs in our GWAS. Okay, so I see there's another question. Professor, how do we interpret linkage disequilibrium decay in GWAS studies? And what does it tell us about mapping resolution and marker density? Yeah, so let's see. How do we interpret linkage disequilibrium decay? I think, okay, in a nutshell, the best way to measure linkage disequilibrium decay is to identify a peak-associated SNP and then measure linkage disequilibrium in the surrounding genomic region. And actually, stay tuned, Akani, because I'm going to show some plots that's going to answer this question directly about how to use linkage disequilibrium in a GWAS result. All right. Um, so let's see. Oh my gosh, here we go. Okay, so first thing we want to do is see how good of a job our model does of accounting for population structure, all right, and familiar relatedness. So what we have here is a QQ plot. Our x-axis is the expected log transform p-value under the null hypothesis. And sorry, I'm on a new computer, so my mouse is always changing slides on me. Yeah, and let me just ask really quickly. Let's see, so we're good on time. Like if in case we go over the 1 hour time limit, is that okay? Or should I step on the gas pedal and try to finish this? And
It's fine, Alex, if you go over the 1 hour period.
okay. Okay, cool. Then cool. I will relax then and just go over this. Okay, so let me ask you all a question. Does anybody know what distribution p-values have under the null hypothesis? Anybody? Okay. So here's the underlying statistical theory of this plot that we're looking at, okay? Under every single null hypothesis in the entire world, if it is true, your p-values are going to follow a uniform 0, 1 distribution, okay? And taking the jargon out of what I just said, under the null hypothesis, a p-value is equally likely to be any value between 0 and 1, okay? So with this QQ plot over here, okay, we are testing to see how closely do our p-values follow the theoretical distribution under the null hypothesis, okay? And taking a step further back, I don't know about you all, but it seems to me that most of my markers, I would expect the vast majority of my markers in my data set to not be associated with the trait, right? So that is, I would expect the null hypothesis of no association to be true for the vast majority of the markers that I'm testing, right? So here we're plotting our observed transformed p-values against our expected transformed p-values. And if these blue dots lie along this diagonal line, that's going to suggest there's evidence that the p-values follow this distribution under the null hypothesis, right? So here, I'm seeing a bunch of blue lines along the diagonal line, suggesting the null hypothesis holds for these likely. And then, oh my gosh, my mouse. And then I'm seeing 3 dots that are veering off. So maybe these 3 dots are in linkage disequilibrium with some, biological genetic mechanism that's controlling our trait of interest. Okay? So anyways, and if none of that made sense, this is a QC check. If most of your blue dots are along the diagonal line, your model's probably doing a good job of accounting for population structure and relatedness. Now here is an output from GAPIT, okay? What we have here is all of our SNPs sorted from most significant to least significant. So we got our p-value, okay? And we see this peak association on chromosome 5, okay? So we have our SNP name, our chromosome position, our p-value, our minor allele frequency, number of individuals, and then we have a, let's see, and then we have our Benjamini-Hochberg false discovery rate adjustment for testing for statistical, adjusting for multiple testing. So if this p-value is less than 0.05, then I would say it's statistically significant, and we reject the null hypothesis. And then this is the allelic substitution effect, okay? And this is measuring the expected increase in our trait value for each additional non-reference allele at this gene. Okay? So this, oh, okay, so this 0.399, that is the estimate of this, okay? And xi is this gene over here, and this is the unified mixed linear model. So any questions about this summary I went over? Okay, let me look at the chat. Okay. Oh, and Nelson, thank you for sharing the Don't Blow Up Twice paper. Um, okay. Okay. So, okay. Now, here
Sorry,
is
sir.
a,
So statistically, you are saying this particular SNP is what is responsible for the vitamin E allele.
sorry, can you say that one more time, please?
So are you saying that that first SNP, the one that's significant, is the marker associated with that trait, the vitamin E?
Yeah, yeah. So what I'm saying is this. So we have this marker over here, like PZB022A3.1, right? Um, so we can say, let's see, Nelson on Thursday was talking about, you know, doing the QC processing, right? So you have 0 for one homozygous state, you have 1 for the heterozygous state, and they got 2 for the other homozygous state, right? So, what this allelic substitution effect estimate is saying is as you go from 0 to 1 or from 1 to 2, okay, the expected increase in the trait value is going to be 0.3393433. Does that answer the question?
No, I know about that effect, but that the SNP itself is the allele associated with is the marker associated with the traits that we're looking for, because I think GWAS tries to identify the nucleotide itself, the very QTN responsible for the trait.
Okay, so sorry, you are asking, is this SNP causal?
This is the causal SNP, or causal polymorphism we're looking for.
Right. So no, this is a great question. Yeah, we have zero evidence here from this table of results that this particular SNP is causal. So yeah, so a GWAS cannot, you know, you know, assign causality to our associations. This is a pure statistical analysis. So this is just telling us that this particular marker has a statistically significant association with the trait. Does that answer the question?
Very good, thank you. Yeah.
You're welcome. You're welcome.
So what can we then say is the causal trait, or I mean the causal marker? What can we use to what can we associate that trait with now?
Right. So you're asking what can we do for assigning causality based off of this result? Is
Yes.
that correct?
Yes.
Yeah.
Yes.
So follow-up analyses that do not involve GWAS should be conducted. So, for example, for this particular association, my colleagues, or sorry, a different lab, for example, they went into the laboratory and they did like a wet lab experiment where they, measured RNA-seq data, right? And so then they were testing the hypothesis that maybe this gene has is associated via RNA, or this like gene that it's near follows. Oh my, I'm I'm not doing a good job of answering this. Let me back up. So you can do follow-up studies to validate this statistical association, right? So you can do a follow-up experiment that measures to see whether or not, the gene this is tagging is controlling a trait through RNA. You can do like a knockout mutation study to see how the phenotypes look different if there's a knockout. Um, but in the ensuing slides, I'm going to show a follow-up statistical analysis we did to give really strong evidence that that the gene that is nearby this association might be doing something. And the take-home message of this long-winded answer is that a GWAS cannot designate causality. Follow-up biological studies need to be conducted. All right?
Yeah, great. Thank you.
Okay. So, okay, another quick display. We have a Manhattan plot. So this is I believe that this is from a subset of our markers that's going to be part of our activity on Wednesday. The x-axis is chromosomal position in maize. Y-axis is a negative log base 10 p-value. Okay, oopsie daisy. And then this is the peak association that we saw. So PZB022A3.1, that's this over here. All right. Okay, so there was a question about linkage disequilibrium, okay? And what is the most effective way to study linkage disequilibrium? And then I said what you should do is measure LD with your peak SNP. And that answer deserved, or this is such an important question that was asked that I made sure we had a slide visualizing this so it makes sense, okay? So here is a plot, all right? Now, the x-axis is position along the maize genome, okay? And we have two y-axes, okay? This y-axis over here, that is our negative log base 10 p-value, okay? And then you see these blue versus gray lines, right? These lines are depicting the negative log base 10 p-values, okay? So here, this is PZB022A3.1, this peak associated SNP. The blue line is the tallest, so that is the most significant association, okay? Now this dashed line over here, this is the negative log base 10 p-value of the least significant SNP, okay? So all of the lines that are blue are statistically significant. All of the lines that are gray are not statistically significant, right? And let's see. So this is the approximate position of the candidate gene that we knew about from prior studies. Okay. Now let's see. And then here are some other potential signals. So we're seeing that another thing, Alex. So here's where I'm going to talk about LD. So these purple triangles are measuring LD with this peak SNP over here. Okay. So let me ask really quickly, are you all comfortable with the measurement of R squared? Okay. So essentially we are measuring the Pearson correlation between the peak marker and all of the other nearby markers. We're measuring that Pearson correlation and we're squaring it, okay? So the larger the R square values, the stronger the linkage disequilibrium is, right? So I see this marker right next to the peak association, right? This purple triangle is over here at 0.75. So this is in strong LD with this particular peak marker. And then all around here, we see purple triangles that are a little taller than throughout the rest of the genome, right? So this is saying that these markers all around here are in linkage disequilibrium with this peak-associated marker. And then we see, okay, LD is breaking apart because I'm seeing that the triangles are getting lower and lower and lower. Yet we're still seeing a blue line here and a blue line here. So LD is breaking down, but we're still seeing statistically significant associations. So we conducted a follow-up study that I'll elaborate on in a second to try to get a picture of what's going on. But the person who asked about linkage disequilibrium and how to quantify it in a GWAS, here's my answer to the question. Are there any follow-up questions to that? Okay. And let me check the chats. Okay. All right, so this is what we did. We're seeing short-range LD decay with the peak SNP, yet we're still seeing statistically significant signals, like those blue lines we see. They're up to 4 million base pairs away from this candidate gene. So what we did, and let me hide my floating media controls. So one of my favorite multi-locus GWAS models is the MLMM from Segura et al. So I'm going to go over the bells and whistles of this in a second, but it's essentially the unified mixed linear model that also considers multiple loci. So let's see. We've got our phenotype of our ith individual. We have, a bunch of markers that are selected through the model selection process. And these multiple markers are encapsulated by this over here. And then we have a subset of markers selected from stepwise regression. And then we have fixed effects account for population structure and random effects account for relatedness. And we also have our grand mean and a random error term. So what we did is we took all possible SNPs on the same chromosome that this peak association was on, okay? And then we conducted the MLMM procedure, letting it do its stepwise model selection. And then the optimal model contained a total of 3 SNPs, okay? And here's a summary of the 3 SNPs, all right? So this is depicting the chromosome 5 region, okay, of maize. This is the model of our candidate gene, all right? And so here is the peak association we identified, PZB03381.1, okay? And it's in this part of the gene. So sorry, I was saying previously it was near a gene, it's actually in the gene. And then, oh my gosh. And then the MLMM, the multi-locus model included this SNP, and this SNP. So the MLMM is telling us that our signal was explained by 3 SNPs, and we saw a substantial fold change in our phenotype values between the least and most favorable haplotypes. So, there's statistical evidence that these 3 SNPs are collectively doing something. And then here's the punchline of all of this, right? So we were originally, from our GWAS, we were concerned about these significant associations way far removed from our gene of interest. So when we reran a GWAS, including these 3 SNPs as a covariate, check it out. I don't see any more blue lines, right? After we accounted for these 3 SNPs, right? So this is suggesting to us that these 3 SNPs we identified seem to collectively be explaining the long distance significant associations. So what we can do with that results is we can talk to our molecular biologists, functional biologist colleagues and say, hey, we have really strong statistical evidence that this ZMVTE4 gene is doing something for our trait. Can you please conduct a proper follow-up wet lab study to investigate this. Okay, so here's what we did. We talked about blues versus BLUPs. We did an introduction to GWAS. These are essential first steps for quantifying the genotype and phenotype landscape. Tomorrow, we're going to talk about more sophisticated GWAS models, and we're going to do an introduction to genomic prediction. All right. So, are there any questions? Oh, I see that there's something in the chat. Lecture 9, part 2. Yep. Lecture 10. Yes. Thank you, Samir. Okay, well, how far are we over? Okay, so sorry for keeping you here for an extra 16 minutes. Are there any final questions?
If you have any questions, please type them in the chat or raise your hand. Okay. Not then, it's just to say thank you, Prof. Alex Lipka, for this lecture, and we look forward to catching your lecture tomorrow. So a round of applause for Prof. Lipka.
Okay. Thank you, everybody, and I look forward to giving this lecture tomorrow. Goodbye, everybody.